Hello everyone, welcome to another video of this revision series. In this video, we will talk about hemostasis. Hemostasis literally means stasis of hemorrhage, that is stopping of bleeding. It refers to all the physiological process that stop bleeding from an injured vessel. And this whole hemostatic process consists of three consecutive processes. Primary hemostasis consisting initial vasoconstriction followed by platelet plaque formation. Secondary hemostasis, that is the activation of coagulation cascade and formation of clot to prevent bleeding permanently, and healing of tent, that is by cell division and fibrinolysis. Now we will discuss this one by one. Suppose there is an injury to a blood vessel. As soon as there is an injury, there will be sympathetic discharge from the endothelium innervated by these sympathetic fibers, and this will lead to transient vasoconstriction. As the vasoconstriction occurs, it is an attempt to stop further bleeding, but as it is transient, so some more lasting process should be adopted, and thereby the next step, that is the platelet plaque formation, initiates. That is, when there is an injury, there is also exposure of the endothelium collagen, and this endothelium collagen will attract the circulating von Willebrand factor, thereby deposition of the circulating von Willebrand factor on the endothelium collagen, and this will cause the attachment of platelets to the von Willebrand factor via glycoprotein 1b9 receptor. And as soon as this receptor attracts the platelets will be activated, and this activation is by development of pseudopodia, increased synthesis and release of Thromboxane A2 from the plasma membrane component, and we will discuss this later in details. And there will be also release of some chemicals like ADP, serotonin, thrombin, etc. This ADP and Thromboxane A2 will act on their receptors on the platelet surface by autocrine or paracrine fashion, and thereby there will be upregulation of glycoprotein 2b3A receptor on the platelet surface, and the receptor bind fibrinogen and cross-link the platelets. Finally, platelet plaque formation occurs. Up to this whole process is called primary hemostasis. Now, secondary hemostasis or true clot formation will occur. This is by the coagulation cascade. Cascade means sequential activation of clotting factor by the preceding factor, and it is mainly by two pathway, intrinsic pathway and extrinsic pathway, and both have a common pathway from factor 9 onwards. Intrinsic means when there is minor bleeding and the bleeding is up to the blood vessel wall, that is, there is no deep tissue injury, and when there is deep tissue injury, it will activate the extrinsic pathway. And whenever we will test some clotting time like test using capillary glass tube, then also intrinsic pathway is activated and the clotting is due to the activation of intrinsic pathway. So in intrinsic pathway, glass surface or collagen due to the breaching of endothelium, this will the initiator of this pathway, and in the extrinsic pathway, by deep tissue injury, the exposed tissue thromboplastin will initiate the pathway. Anyhow, so in intrinsic pathway, first factor 12 is activated by the glass surface or collagen, and this activated factor 12 now activate factor 11, and activated factor 11 will now sequentially activate factor 9 and 8. Similarly, tissue thromboplastin from deep tissue injury will activate factor 7, and this activated 7th factor will now activate factor 9 and 8, initiating the common pathway. And this activated complex will now activate another complex, factor 10 and 5, and this activated complex is called thrombin activator because it will activate the prothrombin or factor 2 into thrombin, and thrombin will activate factor 1 into fibrin, and it will undergo polymerization with the help of another activated factor 13. As a result of the polymerization, fibrin thread occurs, and this fibrin thread along with the platelet plaque and the blood vessels will now form the stable clot that will prevent further bleeding. There are some congenital deficiency of these factors, like deficiency of factor 8 is regarded as hemophilia A, that of factor 9 is hemophilia B, and that of factor 11 is hemophilia C. Also, there's the deficiency of this factor due to vitamin K deficiency or warfarin used related bleeding is mainly due to the deficiency of factor 2, 7, 9 and 10. And heparin inhibits the factor like 2, 10 and also 9, 11th and 12th. And also whenever there is a suspicion of bleeding, we usually do some test like APTT or CT, which mainly assess the intrinsic pathway, and prothrombin time, which mainly assess the extrinsic pathway. Now we'll discuss the fibrinolytic pathway. When there is thrombin thrombomodulin complex formation occurs, this will active the inactive protein C into activated protein C and protein S. This activated protein C will inactivate 3 factors, factor 5A, factor 8A, and also the inhibitor of tissue plasminogen activator. As a result of this inactivation or inhibition of the inhibitor of tissue plasminogen activator, actually tissue plasminogen activator gets activated, and this will now activate plasminogen into plasmin, which is also called as fibrinolysin, because it will now lyse the fibrin into fibrin degradation product. So this is the fibrinolytic pathway. Now finally we will move forward to the production of thromboxane A2 pathway. Due to the binding of platelet with von Willebrand factor, there is release of some platelet activating factor which will activate phospholipase C, an enzyme responsible for the degradation of membrane phospholipids like phosphatidylinositol or phosphatidylcholine. Like phosphatidylinositol will break down into diacylglycerol and inositol triphosphate, which will sequentially activate phospholipase A2 and to act upon phosphatidylcholine, and arachidonic acid formation thus occurs. This arachidonic acid will now undergo cyclooxygenase pathway to produce prostacyclin or thromboxane A2. Prostacyclin formation occurs with the help of prostacyclin synthetase, and thromboxane A2 formation occurs with the help of thromboxane synthetase. There is also formation of prostaglandin D2 from this cyclooxygenase pathway. So thereby this thromboxane A2 will now activate the platelets in autocrine or paracrine fashion. So this is all about the hemostasis. Thank you guys for watching.